The overall goal of this methodology is to expand beyond the optical limit of most flow cytometers by allowing researchers to record 5 additional markers to interrogate complex cell populations. Using this technique, it is possible to achieve a deep immunophenotyping when working with instruments with a low number of detectors or samples with limited number of cells.
Begin this procedure by carefully transferring drawn blood into a 50 milliliter conical tube. Dilute the blood with an equal amount of PBS without calcium and magnesium. Add 15 milliliters of density gradient medium to the bottom of a new 50 milliliter conical tube. Carefully overlay the diluted blood on top of the density gradient medium, avoiding any mixing between the density gradient medium and diluted blood. Centrifuge at 400 times G for 30 minutes at room temperature with no break to avoid disruption of the interface. After centrifugation, use a pipette to carefully aspirate and discard the upper layer, paying attention to not remove cells at the interface between the plasma and density gradient medium. Collect as many peripheral blood mononuclear cells, or PBMCs, as possible from the interface without touching the red cell pellet at the bottom of the 50 milliliter conical tube, and transfer to a new tube. Add PBS to bring the final volume to 25 milliliters and invert several times to mix. Then wash the PBMCs twice as indicated in the text. After